Swayam Prabha. Digital India. Educated India.
Okay, ladies and gentlemen, I think we can continue a little bit. Okay, so there are a couple of things that I would like to tell you before we start with the presentations and so. One of them is that I would like to know more about you, and you know, I don't even know what your name is and who is who. So I would like to invite you, if you think it's all right, and I hope you don't feel uncomfortable, that we make a kind of tour. And kind of introduce yourselves for a couple of minutes or whatever period of time, just to know who you are, where you come from, what is your background, why you think that these topics could be interesting to your current activities or your future endeavors. So, you know, given that, you know, as I said at the beginning, we want to run this course as a kind of conversation. I think it's good that we know each other. And those that eventually remain in the group, eventually, in my opinion, should maintain some type of cohesion. And the cohesion is that you have all participated in this course, right? So I think that it's good that we start some kind of activity of knowing each other and see exactly who you are and where you come from and all the rest of it. Would that be all right? Would anyone feel comfortable doing that? Okay, great. So I'm going to take a seat. Okay, so where should we start? We will start randomly. Well, maybe we can start clockwise. So you are the newcomer, so please tell us about yourself.
Can I introduce my name?
Sure.
So the name I wrote there is not really the name I say, like there, because my name is a much larger one. This is like short one.
Okay.
My name was Shonko Nabo Ghosh.
Okay.
So I say the first part of the name as Shonko. Shonko.
Please don't talk to me.
So I'm Shonko. Hi everyone. I'm a second year bioengineering student here at IIT Madras, and what else? I joined. So initially I did not know about this workshop. I got to know about it from my friends here, and I realized that there were topics taught here that are very interesting, that I find very interesting, and I really want to get into not synthetic applications of synthetic into immunology and pathogens. So I decided that this workshop would be really good for me. So that's why, even though I'm like 2 classes late, I really pushed hard to come here, and yeah, here I am. So thank you for giving me this opportunity as well. And yeah, that is it. I guess.
Okay, thank you. Thank you so much.
Hi everyone, I am Asha and I am in the second year in the Biological Engineering course here at IIT Madras. So I have enjoyed biotech for a long time. Like I had been intending to go into biotech for a while, even like before 12. And I thought that like getting some exposure to synthetic bio might be interesting, since I want to kind of expand like my avenues in terms of my knowledge of biology. So yeah, looking forward to the rest of the classes of the course as well.
Okay, thank you.
Good afternoon, everyone. My name is Pranti. I'm doing my biological sciences undergrad course here at IIT Madras. And I took up this course because I've heard about synthetic biology, the basics of SynBio, because I'm part of the iGEM team for IIT Madras. And I wanted to learn more about it because I found it interesting when I worked on it very briefly last year. So that's, and
Okay.
I'm finding it very interesting. Thank you, sir.
Okay, thank you.
Hello everyone, my name is Tejas and currently I'm in MTech first year, Bioprocess Engineering. I got to know about this course from my batchmate Muthu. I'm interested in genetics and I thought that for the next year MTech project, I may be able to explore this field and apply it in my MTech project. So that's why I joined this.
Hi everyone, I'm Muthu Arunachalam. I am currently pursuing my first year Masters in Bioprocess Engineering. Generally, I was fascinated about how biological systems can be manipulated uh, to get our own thoughts incorporated into them. So I thought that this is one of the platforms through which I can understand and do experiments based on that. So I came here. Hi, I'm Aditya. I'm also from second year biological sciences in Bachelor's Pranati, and yeah, I'm also like very interested in more about different fields of biology, and I think SynBio offers a lot of scope for more fundamental understanding also, as well as like it's a large area for uh, applications, and it can has a lot of potential essentially. So I thought it might be a very good opportunity to like learn more in this field. So I joined this workshop. Yeah.
I'm May. I'm doing my second year undergrad in bioengineering here at IIT Madras. I joined this workshop because the concept of making circuits using DNA sequences and the whole concept of genetic circuit fascinates me. And I wanted to get to know more about that and how that can be applied.
Hello everyone, I am Abhigyan and I am a BS in data science student at IIT Madras. I have a background in biology. I have done my BSc in physiology, and I am basically interested in the overlaps between data science and biology, and that's why I am here.
Sorry. Hi, I'm Aditya. I'm Aditya Ray. I'm also a second year student in biological engineering. So the reason why I took this course is essentially SynBio is a way of looking at biology in like a different perspective. And I think that's a really important sort of thing to have, looking at every single instance from all available perspectives that you can. And this is just something that I've also been really interested for a very long time. So just pretty excited to be here.
Hi everyone, I'm Pooja. I'm also doing my second year in bioengineering, an undergrad student. So the first time I heard about like synthetic biology was in my first year here, and I was very interested in it, but I didn't really have an avenue to explore it. So I was really excited when this workshop came up because I really wanted to learn more about SynBio. So I'm very enthusiastic about what's going to come next in this course.
Hi, I'm Vaishnavi. I'm a PhD student working in the field of metabolic engineering and synthetic biology. So basically, I'm using a strain called Pseudomonas, which Professor Lorenzo is a peer in it. I've used some of his strains and systems for genome editing of the strain for application in the field of lignin valorization, that is converting a waste biomass to value-added chemicals. So I'm looking forward to learn a lot of new things from him. Yeah. Thanks.
Hi everyone, my name is Sachin. I'm third year undergraduate student at IIT Madras in biological sciences. I'm very interested in genetics, synthetic biology, and neuroscience. So I know in genetics I learned from genetics from my courses in neuroscience from this semester when I took course, and I will start my project in neuroscience. But I was always fascinated by synthetic biology, how we use the biological application in real life, how we can synthesize circuits, how we can use to like serve as creating a leather without killing animals, so we can help others. So that's why I took this, and I'm very interested in this course, and maybe I'll choose my future in synthetic biology.
Hello everyone, my name is Devamata. I'm a PhD student in Dr. Karthik Raman's lab. I work on genetic circuit design. So I'm trying to find out computational frameworks which would enable us to computationally design genetic circuits before they are taken to the wet lab. So I have a background in VLSI design. So I felt that the vision that we get for chips where we already have abstractions and everything sorted, and people have generated chips like in a very, very large scale. I feel that vision could be somewhere ported to synthetic biology. And hence, I am just looking for such opportunities where I can apply some of the knowledge I have from chip design to synthetic biology in whatever way it fits. So I am hoping to learn a lot through this workshop. Thank you.
Hi friends, I am Abdul Rahman. I am from second year undergrad in biological engineering. I took this course because I thought it is fascinated and I am a little bit interested in neuroscience, and I thought that it is more related to the synthetic bio also, like logic gate and those things. That's why I took this workshop.
Okay, well, thank you. Okay, so it seems that many of you come from second year bioengineering, right? Okay, so how many years bioengineering is? 5. Okay, so it's a whole career in bioengineering. Okay, very good. Okay, great. So we know what we are, what kind of human material we have. Wonderful. Okay, great. So that's one thing. So I really hope that we can maintain this kind of team for some time with maybe some plus and minuses. If you find, if you know some of those who defected and you can still convince them to be back, they will be very welcome, okay? So we will not wait for them with a tick, just the contrary. We will welcome them and everything will be fine. Okay, so the other thing that I want to start telling you about the plans for the rest of the course is the following. This course is a 10-day course. I mean, we cannot pretend to really cover absolutely everything, but to what I want to deliver is a kind of a snapshot of the different flavors that are associated to synthetic biology. And so this week, as I said, will be quite intensive because I will try to deliver as much as I can on fundamental concepts in synthetic biology. Some of them we have already covered, you know, the first day and the second day. 2 important days because I will talk about another fundamental in synthetic biology, that is the concept of the chassis, synthetic biology. And then there will be standardization on vectors, genetic technologies, genetic circuits, metabolic engineering, and so on and so on. But I said in my email this morning or yesterday, so I would like to spend the second part of every day to do an activity where you guys are involved. So during this week, the activity will be presentation of papers, and I hope that in the next few days, you know, we have the 4 presentations per day rather than today that we will be like half year. But then for the last few days of the course, what I would like to propose is the following, and I want to give you some advance notice of what I want you to do. So one of the many flavors in which synthetic biology comes is through this thing here, what is called the iGEM. And one of you just mentioned that, so maybe for you, I don't have to explain what it is, but it goes as follows. In the early, some historical perspective. So in the year 2002, 2003, there were 2 people in the MIT, one of them called Tom Knight and the other called Drew Endy, that in fact, you know, came up with this idea that biology could be the subject of some serious engineering. And one of the first ideas that they had, and this was mostly the idea of someone called Drew Endy, that now is an entire celebrity, if not a kind of saint in the whole picture of synthetic biology, is that he developed this concept of biological parts, as I said the first day. So these parts, with his engineering perspective, said, well, a part is like a brick, right? So let's call these parts a biobrick. So a biobrick is a piece of DNA that contains or encodes a biological part, but is formatted in such a way that it can be assembled easily with other parts. I will give you details much later because in one of the classes I will discuss that in detail. But just a kind of advance. So he started to make a collection of so-called biobricks. So, and biobricks is a collection of biological parts, each of them with a defined function and also with a defined code, like, you know, part number, this and that, as you have with parts for cars and other man-made devices. And he said, well, what can we do with the parts? How can we start people getting interested in using these parts for doing useful things? So they say, well, I'm going to propose the following. Let's make a competition and let's propose undergrad students of biology, of engineering, to get together during summer, and then to use these parts as a kind of Lego for building biological machines. So it's called engineer machines. Okay, the M goes for machines. So then it started like a kind of little competition in the Bay Area around Boston, MIT and Harvard and so on. And then the idea, as I said, was to propose universities to form teams, mostly between engineers and biologists, to get together to work during the summer, and using these standardized parts, these standardized biobricks, to make constructs that could be useful for something interesting. So this started as a small endeavor, and then with the time, it attracted a lot of attention. And the reason for that is that when you let young people, mostly undergrads, to propose, to be creative and to propose applications for this type of approaches that I have shared with you these days, they can be incredibly, creative and amazing in the type of things that they propose. So, so they said, well, this is a very good start. Why don't we propose to amplify the community of, say, IGM participants? So over the years, this community has grown and grown and grown to the point that now it's a super big endeavor. And the last meeting that took place in Paris, had like thousands of participants. And there was a team of ITT that, by the way, I don't know who they are, but at some point I would like to meet them, given that I'm here. But I don't know. Does anyone know who these guys from IIT are, or where do they come from? This. Okay, so you think that you think that no one in Chennai participated in this IGM thing? Yeah. Okay. Well. Yeah. Okay. That makes sense. Well, in any case, so the deal has plus and minuses, because what started as an academic endeavor has ended up like a kind of big business. Goes as follows. So one university decides to participate in this IGN competition, and then the idea is to identify biologists and engineers that get together, maybe a team of 5-10 people. And then they discuss a project, that the project has to follow some, guidelines. One of them is that all constructs have to be made with standardized parts, with biobricks. When the IM competition started, there were just a few biobricks. Now there are thousands of biobricks. And if the biobricks are not there, then you can propose to build a new biobrick, and you have to follow a given format. And then once you have it, you send it to a repository of biobricks that is kept somewhere. At the beginning, it was MIT. Right now, I don't know where it is, but still, you can get biobricks for different applications from the IGM organization. In parallel, there was this other thing that is called the BioBrick Foundation, that is a kind of more academic, a branch of this whole movement about the standardization, biobricks, synthetic biology, and all the rest of it. Okay, so then the different teams they have to register and they have to pay, and the fee is not cheap, so it's a considerable. But many universities eventually sign up because it's a kind of prestige that you participate in this IGN competition. And then during the summer, the teams propose to construct something that is useful under the idea of synthetic biology, and they have to do various things. They have to develop the concept, to develop the structure of the possible constructs, to assemble these constructs, and to the best of their possibilities to check them in terms of activity. As these projects are summer projects, in many cases they don't get to the end. Okay, so this is more a kind of training exercise than a real application. But the most important part is the conceptual part. So they can propose a large number of things, I will tell you a few of them that to me were quite nice. And then at the end, there's this called a jamboree. You know what a jamboree? Jamboree is this idea that Boy Scouts get together and make a big party or whatever, and then from all over the world. So they have these iGEM jamborees that for many years had taken place in Boston. Now they will be, for the next few years, will be in Paris. So different teams will get together and there will be a competition. So a competition means that each team will make a presentation about their propositions, their intentions, their motivation, their parts, their circuits. And there will be a jury that will listen to the different presentations and will make a qualification. And at the end, there's a jury, it's a competition. At the end, people get different prizes or get nothing, but there are many prizes, like the prize to the best part, the prize to the best concept, the prize to the best device, the best system, the best. So at the end, there are many prizes. So people get more or less happy. It's not only the Oscar goes to just one movie, it goes to many different things. So what is the advantage of this approach? Well, the advantage of this approach is that people are encouraged to be creative. And because there's one phenomenon that as you progress in your career, you will notice as you become biotechnologists, and is this bloody issue of intellectual property. Okay? So intellectual property, that means that, I'm very smart, and then I develop an expression system that is inducible by tetracycline. Okay? Okay, very happy. And then my employer say, well, this is very nice, but I want to patent this. And if you patent something, that means that now if everyone wants to use it, either they have to pay or at least they have to keep you informed of what you do with it. And the ones that get the intellectual property keeps a degree of control of this particular part, okay? This is something that is very good for the owner of the intellectual property, but it's very bad for the rest of the world, because if I own one thing for myself, I avoid that you develop it in a different direction, okay? So part of the philosophy or the motivation of iGEM is to have a completely free thinking on things that are done under an entirely open, ecosystem for creativity in synthetic biology, okay? So you ignore intellectual property, you ignore anything. You just go to the list of parts and you don't care whether the parts are protected, they are not protected, you don't care. And that's something that in many cases only undergrads can handle, people like you, like many of you, because you are not aware and you are not constrained by all these limitations on intellectual property and these things. So you just think free and then you propose something independently of what will be the string consequences. Okay, so that would be one thing. One other aspect of the whole iGEM endeavor is that there's always an eye on societal consequences. And one of my, by the end of this week, we will have, well, not by the end of the week, but on Saturday, those of you that want to come on Saturday, we have a little seminar on legal, ethical, and societal consequences of synthetic biology. And part of the iGEM package is not only to describe the devices and the applications and everything, but also to make a study on the societal consequences of your, proposition. Obviously, the societal consequences may come a bit later, because in the period of time that you are under this program is too short to really have societal consequences. But there's this idea that one has to study and to propose, keep an eye on the entire life cycle of the constructs that we want to develop. So that would be the frame. Okay, so in this course, like a iGEM team, because we don't have the time, we'll have the money, and I will go at some point. So that's something that is out of the question, even though maybe at some point you can propose your superiors and your professors perhaps to put together a formal iGEM team. But I would propose you is the following. The last days of the course, instead of having this paper discussion, because by that time we have already talked and everything, what I would propose is that we make a kind of IGEM competition in which each of you go to the IGEM, say, repository, take a look to projects that have been done by people there. And obviously, you don't have to imitate what they say, but you get the kind of feeling of the things that you may propose if you were given the opportunity to construct with the tools of synthetic biology something for a given purpose. Okay. So what I would like to propose is to form a kind of IGEM teams that will not be identical to the teams that we have made for paper reading. So we can form, if you wish, 4 IGEM-like teams, and then you just go through the different information, and then you start thinking on what type of, I will not say crazy debate, but what kind of construction would you like to propose for discussion and viability studies. Okay? So what are the typical, some typical IGEM projects? Okay, one IGEM project, in fact, I told you in my first talk, was this idea of converting E. coli into a photographic film. So this paper that eventually ended up in Nature or in Science, I don't remember, Nature project, because it's a kind of crazy thing to entertain graphic film. So that was one of the very earliest. But there are others that were quite interesting, and some of them are, in fact, have been followed to some extent to be actual biotechnological issues. I'm going to tell you a couple of them, if I can. Yeah. I'm going to make a picture of this, by the way. Okay, now. Okay, this does not work well. So you think that if this were wet, it will work better? Yeah, because you know. Can you put some water on it, maybe? Okay, well, one of them that was even made it to the headlines in press was the so-called Dr. E. coli. I don't know if you ever saw that. Is it better? Yeah. Yeah. Was much better. Okay, great. Okay, so this Dr. E. coli it went as follows. So imagine that you have an E. coli strain that you engineer to express different colors depending on environmental signals of some chemical nature. So if you, for instance, have a disease and then the disease results in some inflammatory response, then the response is associated to production of some small molecules for which you can develop a biosensor. And then the biosensor can be coupled to production, I don't know, of green protein. And then you may have another biosensor associated to production of a diabetes marker. And then you can have another color that is associated to production of, say, a small molecule associated to a specific type of tumor, for instance. I mean, you can make a list of diseases and then a list of small molecules that are associated as indicators of that particular disease, okay? Now, you make the construct and you make this circuit in which you have E. coli with all these different, thank you very much, with all these sensors, and then what you do is that you give these sensors as a pill to the patient, right? So this bacteria will be in your gut, will eventually colonize your gut, probably, and at some point you will secrete your depositions, and then the depositions will have a color, okay? So by looking at the color of your feces, you may immediately have a diagnostic tool telling you whether you have some important health problem, okay? So that could be, there's a picture that I don't have here. I will show you when I talk about the IM thing more in detail, where you have like different pieces of different colors, and that is an indication that you have this or that disease. Obviously, to do that, you require a lot of concept on the type of circuits that you have to put together, the type of say, reporters that you have to put together. You have to examine durability. I mean, you have many issues that are typically connected to the science of synthetic biology, but at the end you have something that is really useful for this type of application. So that's one project that made headlines at some point and is very, very good. But you can do something completely different, completely different. So one project that I found super creative and has nothing to do with biotechnology, it will be the following. There's this article or this project by a Japanese group in the mid-2000s, or I think it was like 2010 or something like that, that it was to recreate with bacteria the Romeo and Juliet story. Do you know Romeo and Juliet? Okay. Okay, well, Romeo and Juliet is a Shakespeare play of two lovers, two young lovers that live in Verona, in Italy. They were young, they were teenagers, they were in love and everything, and their families didn't want them to be together. And then at some point, one of them takes a drug that appears him to be sleeping, and then she, or vice versa, believes that the other person is dead, and then she commits suicide. And then, well, you have a kind of play that, you know, you have two main characters. The two characters are influenced by the environment, and then one of them develops, I mean, they start developing say, falling in love, and then this falling in love becomes destructive because one of them appears to commit suicide and the other does not commit suicide, and at the end, it's a tragedy. I mean, Romeo and Juliet, at the end, everyone is a big tragedy. But, you know, it's one of these Western icons, you know, Romeo and Juliet, this love that people have when they are teenagers. Okay, so this Japanese team said, well, let's try to figure out how this works in bacteria, okay? So at the end of the day, what is falling in love, okay? Well, falling in love is that, you know, we are separated, but then the more we get closer, we start exchanging messages that we like each other, and at the end, we really get together, right? So they say, well, why don't we take this strain and make it auxotrophic for methionine? And then, but at the same time, you secrete serine, for instance. But then, on the other hand, I have this other bacterium B, where I do just that's the opposite. I make it to be an auxotrophic of serine, but then it secretes methionine, okay? So you can maintain them separated when the environment is the adequate one, but the moment that you remove these amino acids from the medium, the only way to that they get together, that they survive, is that little by little they start exchanging these things, right? Okay, so that's the first chapter is that falling in love. So they get together because of this chemical attraction, right? Number two, the problem. Then, well, what if, you know, I make one of these partners in the interaction, we engineer things in such a way that the moment that the two cells get together, they activate, for instance, a type 6 secretion system in such a way that the moment that there's a contact, then bacterium A injects in bacterium B a toxin, okay? And then you have a problem there. But then this toxin perhaps can be rescued by the action of someone from the outside that produces a quorum sensing system in such a way that when the whole family gets together, you inactivate the action of, I mean, I'm just, you know, not telling you the details, but this is the type of things that these people were able to put together. And I found it completely imaginative and super nice because, you know, it looks like a joke, but it's not a joke. I mean, there's a lot of science and creativity behind that, right? And also, you know, just keep in mind one thing. In synthetic biology, we play a lot with games. So games are something that you artificially put together to discover the rules of interaction, okay? So biologists like models. Synthetic biologists like games. In a model, what you do is that you try to capture the main components of a complex system and you try to make them simple, and then you start playing with them to see how it behaves, to get some insight on the biological system as it is. In a game, what you do is that you take things from completely different parts and play with them. And by playing with them, you understand and get some insight on how the system works. So that's something that is very typical of IM proposition. There's another one in which I was myself involved, and is the following. Can we talk to bacteria? Can we talk to them? You know, you have a picture, a flask, and you talk to bacteria, and you get some response to your questions. Okay, so you say, well, this is completely crazy, right? So how can you do that? So what we did was the following. So the questions had to be very, very simple. I mean, you cannot engage in a philosophical conversation with a bacterium, but you can ask them, are you cold? Are you So what we did was to have again a color code, green, yellow, red, and we made a series of logic gates that implied to have an environmental signal in connection to a command that was addition of an inducer. Okay, let me see how it goes. So you remember the AND gate here? Okay, so you have here a voice. So the voice goes to an Arduino. Well, a voice decoder, Arduino, valve, and then the valve goes here, and then it goes environmental signal. For instance, cold, for instance, heat. And here you have a red. Okay, so then you have this circuit, okay? You have an AND gate that you structure genetically, where you have one of the inputs is an environmental signal, like heat. There are many promoters that respond to heat, but the other is an inducer that is added to the medium when a valve is open, that is controlled by an Arduino device that responds to a voice decoder, and then there's a voice that interprets the question, okay? So if I say, are you hot? Then the decoder will interpret the word hot. We will send instructions to Arduino. Arduino will open a valve that will add an inducer that together with high temperature will give you a red response, okay? I mean, this is something simple. And by combining this with a toggle switch, I can have the same, and instead of red, I have blue here. Okay. So you can say, well, are you hot? If it is hot, the answers, the cells will turn red. If they are not cold, if they are not hot, the cells will turn blue. Okay. So, I mean, this is something you say, well, this is a joke, right? You don't talk to cells. However, you know, from the distance, you know, imagine that, you know, you show this to your family in a family dinner. You go with a flask and say, you tell the cells, are you cold? And then they turn blue. I mean, this is like magic, right? So this is something remarkable. Well, anyway, so this is another project that we were at some point. And more recently, we had in the laboratory something similar here that was like more dramatic. Okay, so one of the spin-offs of synthetic biology, and again I will spend one day discussing this more in detail, is to have what is called artists in residence. Okay, so I don't know if you ever had that experience, but artists in residence is that you are in your laboratory working with your clones and all your things. And then you invite an artist, either a painter or a musician or a plastic artist, or someone with a professional artist to go to your laboratory to spend some time with you, and on the basis of being with you, then they have to propose an artistic project. So we had recently one lady that was a professional photographer from Lebanon, from the Middle East. And then she talked to us and circuits and, talk to bacteria and you do this and Pseudomonas. I mean, she discussed with everyone. And then she said, well, let's do one thing. So we're going to have a bacterium that will commit suicide when you tell them to commit suicide. Okay. Obviously that was very easy, because I mean, this is way more difficult, because all you have to do is to have this Arduino voice decoder together with a valve that will activate a toxic gene. So it's not a super big thing, but but from the external point of view, it was super impressive that you tell bacteria commit suicide, and then the word suicide immediately gets transmitted into something that goes into that type of thing, and cells die. How do you know that they die? Very simple, because the starting point were cells that were luminescent. Okay, they emitted light because we had Lux genes from Vibrio expressed, and therefore the culture was luminescent. I mean, you could see it with your naked eye, and then you talk to the flask and say commit suicide, and then in few minutes the light disappears, right? So that was another case. Okay, so again, these are games, okay, but these are the games that are very frequent in IGN competitions, simply because you can see with these games the power of the technology, because sometimes you can go from playing games to things that are way more serious. Okay, so I don't want to elaborate with that because I insist that I will return to this in more detail in one of the classes, how the IGN is structured and so on. But start thinking, okay, what crazy ideas you would like to share with this group that could be done with some of the technology, some of the tools, and some of the concepts that we are discussing these days, okay? And I'm sure that you will be very creative and will propose amazing things. Obviously, they don't have to do exactly what I mentioned today. I mean, it can be things for applications, for medical purposes, for neurobiology, for for metabolic engineering, for you name it, right? So one topic that is becoming very popular in the last few IGM teams is, for instance, plastics. Plastics. What do we do with plastics? Do we degrade them? Do we do something else with them? I mean, so think about it, and maybe some of the tools that we have discussed these days could be inspirational for you to think on possibilities. Okay, so I close this little sermon on IGEM, and now is the time to go to these presentations that you guys promised to deliver today. We will have only two. But I think that we will have fun in any case listening to them. Okay. So who wants to start? Who you are one of them, and the other, who was the presenter? Okay, very good. You want to start? Sure. So let me just load your presentation.